No hurry? Okay, good. All right. Very good. So let's begin, shall we, with a prayer, and we'll close those back doors and all that in a minute. We're picking up where we've left off, just to give you an overview of where we have been. We were looking at science, faith, and reason, and we're talking about four fundamental features of the world. And the first feature of the world that we looked at together was the evidence for the universe's beginning. We explored that together and saw the steady state theory that was the most common view until pretty recently, actually, which was the notion that the universe just somehow on the fringes of the universe was just somehow being spontaneously generated in order to sustain the universe as it is. And that has now been defeated because of an energy, a singularity, very suspiciously indeed. And this is, in fact, why there was a bias by many astronomers against this very view. They didn't want it. In fact, they spoke of the Big Bang in a pejorative term, in a pejorative sense. Oh, that's just a Big Bang. But turns out, against their bias, it looks like that's how it was. It's not, haven't always been here. And then we looked at a second feature of the cosmos, and we were looking at the fine-tuning and found that it is exquisitely fine-tuned on every order of magnitude, from the microscopic, from the particles themselves and their various ratios, to the macroscopic. And we were looking at that, and we'll say some more about that. And today we're going to be looking at biogenesis, which is the evidence for the beginning or the origin of life. How did you get the first living cell? Here, we're not talking about evolution at all. We're talking, we can't be talking about evolution because evolution requires life, you see, because the whole idea there is mutations and natural selection. But the thing's got to be alive before it mutates and then naturally selects, presumably according to neo-Darwinism. But you see, how do you get the first living cell? And the idea was that somehow it must have been like some kind of in the primordial sludge, something pretty simple and so forth. Turns out that's not the case. It's exquisitely, elegantly complex. The more we learn, every year we're learning more, the more amazing it actually becomes. And then in our next session, we're going to be looking at information theory and the nature of that. So in exploring all these wonderful and remarkable things, we're exploring the fact that there are so many new developments that we've seen that indicate we're just in a just right situation for life itself. We were exploring the fact that we have a just right galaxy, a just right sun. It has to be a certain very narrow range for where we are in our actual solar system, and it actually has to be a just right place within our galaxy itself. In fact, we even talked about a kind of a certain habitable zone in the galaxy. And the more we look at this, the more refined, specific, and fine-tuned it really becomes. So that as a consequence, people have now speculated the idea of some kind of a multiverse, you see, the idea of that perhaps there was some multiverse, that the idea that there was a boundless number of these, to suppose that somehow we now have won the cosmic lottery, you see, because that's maybe why we were. So giving an infinite number of universes and bound then one of them's going to win. This is the idea. It's like when you see the lotto or these big signs and has the number of how many millions of dollars the winner will win. And you can be sure, you can be putting money in that every week and you can be pretty sure you're not going to get it. I remember my father was talking about how, and I was saying, Dad, you might as well just throw your money away. He's investing, as it were. He was doing $50 a week or something like that. You just put it in a mutual fund, you'll be doing better than that. He says, yeah, but somebody's got to win. Yeah, I said, it won't be you. Your chances of winning are better. You'll be more likely to be hit twice by lightning than by that. Somebody will win, yeah, but it won't be you. That's the way it will work, you see. So you got to understand that. It's just the way it works. So the idea is that it gets suspicious, you see. There's no evidence, and this is important, theoretical or experimental, for any universe other than our own. To pose the idea of a multiple universe is often more because of a particular belief system. That is, there's no designer than about science. In fact, it's a step that's outside of science. It's a faith step that has no evidence at all. It didn't say that there couldn't be more universes, but there is no empirical warrant for that kind of speculative idea. This is a construction of mathematics. And so what we're dealing here is we're dealing with mathematical constructs that are highly elegant, and I don't deny that they're elegance. But the fact is, though, it doesn't work in terms of a theory, a system of determining reality. You have to deny the first and second laws of physics, of thermodynamics, the most fundamental laws, principles of physics in order to buy this. As one physicist put it, Edward Harrison, he wrote, take your choice, blind chance that requires multitudes of universes, or design that requires only one. You see, that's how it works. And by the way, what they haven't figured out as well, even if you had an infinite number of universes, it still wouldn't work. You know why? You'd have to have a universe generating machine that would actually be fine-tuned. It had to be as finely tuned to create the idea of a universe as the fine-tuning that you're trying to explain. So it still wouldn't work. So it just doesn't work. And again, they won't let go of it because you see it's one of those in the mountains there. You begin to get rather suspicious that this wasn't a process of erosion and time and chance. You get very suspicious when you see the heads of former American presidents, Washington, Jefferson, Roosevelt, and Lincoln, and of a turtle on a fence post as an analogy to this concept. So when you look at the possibility of a turtle on a fence post, what would you naturally conclude? Let me see if I can find them for you here, because I had for you here. There he is right there. So when you see something like that, have you ever seen somebody do this cruel thing to a turtle? It happens from time to time. But there aren't many explanations for how it got there. Maybe just a storm blew it up there, you see. Maybe it crawled up there. These are not good explanations. You see, it implies very clearly some kind of a design. That is to say, some kind of nefarious design in this instance, where somebody decided to put it up there as a bit of a joke. And that's the concept that's involved here. So when you're looking at something of this nature, then you're seeing that you only have so many options that you have before you. And so we go to this concept then of now our next session, biogenesis. So we're just trying to look at an understanding of the evidence itself. Go where the evidence leads. And that's what we've been talking about. Good science is really inference to the best explanation. Go where the evidence leads. Instead of front-loading your ideas, well, it can't be this. It could be this. It could be chance or necessity, but it couldn't be design. You're now not doing good science because you're not open to the possibility. Remember, we talked about the fact that there are various sciences that actually are doing evidence-based materials that can distinguish chance from design. And if you might recall, we described insurance claim investigation, we described cryptology and artificial intelligence. Archaeology is a great example where you've got to be able to figure out, was this chance or was this design? So it's not like we don't have a category in science for the notion of design. SETI, Search for Extraterrestrial Intelligence, and also random number generation. So it's not like this is something that we don't have there. So we just go, as I said before, going where the evidence leads is where we need to take our place and go from there. So given this notion, then, we go back to our place and pick up where we left off. And so I love this this remarkable dragonfly. That's what it is. It's a little water on the eyes of a dragonfly. And when you begin to study a dragonfly, I recommend, by the way, a couple of places, a couple of good sources for you to look for videos. The John 1010 Project is one. So consider, it's just called on YouTube, the John 1010 Project. They've just posted a new one today. And it's excellent because they're really great, well-produced videos that explore, usually about 4 or 5 minutes long. And then they explore the vast areas of nature itself, many facets. And then there's also the Discovery Institute, and there are a number of videos that are available there and many other places, but this is just to let you know about that. So that it's the idea of the origin of life itself. Now, the cell, there is no primitive cell. There is no simple cell. All cells are exquisitely complex. As Michael Denton observed, that undirected natural processes somehow turn lifeless chemicals into all the extraordinary complexity of living things. That idea, he says, is no more or less than the great cosmogenic myth. It's a myth that's not around. James Tour from Rice University is your key guy on this one these days. I may show you a little portion of what he's describing here as well. But he'd be a great resource for this because he builds these wonderful nano machines, these little motor, like acid-powered motor vehicles, and he makes them run around and so forth. And you have to really be pretty sophisticated in synthetic biology and synthetic chemistry in order to pull these things off. So he knows what it takes to build these things up. And actually, when he talks about, as he concluded, when it comes to kinetic products, time is not your friend, it's your enemy. So just give me enough time, even the most random thing will happen. No, it doesn't happen that way. Unless you, when you get to the next stage in complexity, you need to actually quickly hold that position or it will degenerate, you see. For the next stage, it has to then be preserved or changed or held so that time does not serve you well. And these have to be manipulated. So time does not by itself produce the kind of outcome that you're looking for at all. And so what we're looking at when we speak about the exquisitely complex cell, Franklin M. Harold, in The Way of the Cell, described a single-cell organism as a high-tech factory. It's complete with artificial languages and their decoding systems, memory banks for information storage and retrieval, elegant control systems regulating the automated assembly of parts and components, error, fail-safe, and proofreading devices utilized for quality control. Each cell, approximately 40 trillion cells in your body, is constantly error checking, constantly checking itself and modifying itself in ways we can't even begin to understand for quality control. And furthermore, assembly processes involving the principle of prefabrication and modular construction, and a capacity not equaled in any of our most advanced machines, you see. So this is something that's beyond, for it would be capable of replicating its entire structure within a matter of a few hours. We can't even synthesize a protein. So it just goes beyond our imagination when we consider that. So the so-called simplest cell is far more amazing than we could have ever imagined. In fact, it was previously thought by Darwin and his followers that the body was elegant, but that the cells were fairly simple. So he thought the cells were like Lego blocks by analogy, like kind of protoplasmic blobs, you see, and that they were just kind of nebulous. And so that's what he assumed, so therefore that could be worked out. But it turns out that each Lego, so-called, has the complexity of a miniature body. The more we learn about the cell, the more extraordinary it becomes. It's even more complicated. Any of your cells in your body, really, when fully analyzed, is more complicated than the city of Atlanta. And that includes plumbing and wiring and transportation systems and fabrications and storage facilities and building up and breaking down. They can build super highways and transportation systems, and then it breaks them down after it's used them. Constantly doing this many thousands of times a second. I'll give you an example of this because it's rather fun to give you an idea what this looks like. So if I go to cell biology here, and if you look at your body's molecular machines inside the cell, I'm not going to go into detail here, but just to give you an idea of what this looks like here. You see that this is like a little video that describes parts. These are kind of computer animations of what these parts of the cell look like, and it describes them. But I don't know can we do the sound on this, Keith? Does the sound work or not?
Both proteins emerge from the extracellular matrix, which covers the surface of both cell types. The outer lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the lipid recruit specific membrane proteins.
You're not going to understand what he's talking about, by the way. He's using language that we can barely understand. You'd have to be a real expert to hear it. But I just get an impression.
Against the straight sphingolipids and carbon chains. Uncyclamates, kinks in unsaturated microcarbon chains at lower cholesterol concentration result in increased fluidity. At sites of inflammation, secreted chemokines bound to heparin sulfate protein glycan on endothelial cells are presented to leukocyte transmembrane receptors. The binding stimulates leukocytes, triggers an intracellular cascade of signaling reactions.
You're getting the idea. It's a marvel and a wonder. I will now do the sound, but I'm just going to walk you through just some of the images. This is part of the structure of that cell as it works through here, and these filaments that you work your way through. And these are these severing protein. Look at how it forms these molecules you see in the cell, and it fabricates these nanotubes, and then these are systems that it then tears apart after it's used, and how it then has little things that walk along these vesicles. See the little thing like little legs, motor proteins, you see? He has little feet, you see? And he's walking along there, and he's carrying material along there. And then it goes on from here. So we can't even, this is all within the cell. This is the mitochondria here. And it just goes on and on and on. And so you look at these things, and these are just our ideas of how it's amazing we've been able to figure out this complexity. You see, the more we learn, guess what happens? The more you learn about it, it gets even more exquisite than, not less. So it just goes beyond your imagination. This is
science, mathematics,
it's like fractals and so forth, but it's a combination of things. So this is just one idea of this concept here. Another video here has this idea where there's a fellow who's talking about it here, and I'll let you hear him for a moment.
Tiny molecular machines, and they are doing this inside your body right now. Understand why we have to zoom out? Every day, or damaged, or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look.
I'm just going to give you a sample of this, just to give you an idea of the vastness of what we're looking at.
DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now, the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section.
Work that one out. You see what I'm saying? What is making these things work? Nobody knows. Nobody understands what actually drives them. We're not talking about little motors or muscles or neurons. These are not neurons, you see. It's not a brain. Yet something's guiding these things, and they're happening billions of times a second in your body, all throughout your body, constantly and perfectly, almost perfectly run. And so it just goes beyond belief. Again, I could go on and on with these ideas of what they're trying to describe. And so what this description of these complex wonders and marvels that you see, where these, it forms, walking along there again. And so it just wonderful things to behold. And then again, this is another image here of a molecular visual. Again, the DNA wrapping here. And so just consider how it wraps. Just the DNA, the double helix structure is exquisite. But then it wraps itself, and it creates this wrapping by means of this special protein molecules, the histones. And then this combined loop then works together and creates a nucleosome. And then consider the nucleosome is packaged into a thread, and then it becomes a chromatin, you see. But wait, there's more. And so then the chromatin then is looped another time yet, another coil, and then it's woven together in another level of complexity. You see how this is working there? And then from there, it moves in this complex thing into familiar shapes known as the chromosomes, which are used in mitosis and cell division and so forth. So it's just, again, beyond our imagination, and again, these processes. So I'm showing this to you only to illustrate the concept. We're dealing with something, the more you learn. So we have our models. Amazing how we've learned so much, but the more we learn, the more questions are raised, because we don't even know how this is working. And all these things are happening all at once. So it is wonders within wonders within wonders. It's not simple. This is my point here. And so you're looking at something that implies, if it implies design, so be it. Thomas Nagel, again, he's an atheist, he's a philosopher, but I appreciate him because he's at least open to possibilities that are outside of the normal materialistic province. For a long time, I've been skeptical of the claims of traditional evolutional theory to be the whole story about the history of life. This has made him, therefore, not exactly endeared by many people in that community, but he's just being very honest about the matter. And he says it potentially can be scientific to argue that the data of DNA in life points to an intelligent designer, even if science cannot tell you the identity of the designer or what is going on in the designer's mind, you see. So it doesn't mean that science has to be wedded to materialism. In fact, the more you learn about it, the more it's impossible to account for the mysteries we behold that transcend our imagination and our understanding. Codes for proteins and the development of that and the formation of those, but we don't know. That creates the parts. It's like thinking of a warehouse. It creates all the parts you need. But what assembles the parts into a body plan? You see, it's one thing to make these exquisite, complex parts. Suppose you're going to make a 747 and has all the parts that are necessary in this warehouse. What then takes all the parts and puts them together in the right sequence? Nobody knows. It's called epigenetic information. That's because we don't know what it is. It's found in embryology. And suddenly, because any one of these cells can then become one of 200 different kinds of cells. Once it becomes that kind of cell, it can't then change. And then it changes, it forms, it finds a part in its structure, and it becomes a part of this structure or that. And beyond astonishment. The more we're learning then, the more marvelous it becomes. So what would it take then to get one living cell is the question. You cannot appeal to neo-Darwinian mutation and natural selection, as I said before, because that only applies to living systems. You have to go back to chemistry and molecular biology. Darwin's Black Box, Michael Behe wrote this book a number of years ago. And he's written more since then, and because the evidence gets even stronger as the time goes by. He called it Darwin's Black Box. Darwin, the cell was a black box was a blob of protoplasm. So box, a little bit more, because we now have things such as X-ray crystallography, and we have nuclear magnetic resonance, and we're actually able to delve deep enough into the cell and find out it is really more complex than a city. So as I said before, you have structures that go beyond human engineering and technology. We've never been able to design anything remotely like these things. Oh, we have impressive machines. I don't doubt that. Our AI and our computers are impressive, but they're child's play. And anytime you try to retro-engineer anything in nature, it's always inferior. It's like a child, say in a nursery, trying to imitate Michelangelo's David, or this sort of a thing. It just doesn't work. And so it's there, but it's not impressive. So we're seeing an elegance then that is so wonderful. It's an exciting time to live, because in the last 4 or 5 decades, we've seen a plethora of new things that we could have never previously imagined. And that's what's wonderful about it. I'll just mention proteins, just a word about that, and you could just go on and on about this. But the possibility of creating and synthesizing a simple protein through undirected natural processes borders on the absurd. In fact, this has finally been admitted more broadly, and they finally now plump for an RNA world hypothesis instead, because they've given up on the protein synthesis model. But that won't work. That's introduces its own problems. But with the proteins, though, you're dealing with hundreds of amino acids. And this requires a gradual buildup of a polypeptide bond in precisely ordered sequences. And there are 20 basic kinds of amino acids that we have. And so when we look at these amino acids as well, that's hard enough to get these polypeptides that bond together, because each new one to get is harder than the one before. And there's reasons why that is the case. But even if you grant that, it gets even worse. The reason why it's worse is because there are right and left-handed versions. You see, and it turns out that even the correct sequence, if you got them, it's not only to get them to bind, but then it has to be in the right order. And then third, they have to all be left-handed. Why is this? No one knows. Because you see, each of the 20 different types of amino acids present in the composition of proteins is left-handed, but yet in nature, they appear about with the same amount in regularity, you see. So you can have right and left hands. They're mirror images, like your right and left hands, you see. And so what you have here, both these two types can easily bond with one another, but the astonishing fact that has been revealed by research is that all the proteins in plants and animals on our planet, from the simplest organism to the most complex, are made up of left-handed amino acids. If even a single right-handed amino acid gets attached to the structure of a protein, the protein's worthless. It will not work. So you've worked that one out. How did you get this? And I'll choose a small protein of only 400 amino acids. Some of them are thousands of them. It's like, imagine then, how did you get this? Somewhere along the line, it'd be like tossing a fair coin 400 times and getting heads every time. And it just becomes more and more difficult. So people have to begin to realize that this isn't going to work. Nobody knows why they're all left-handed, but it is. And then it gets even more difficult, because you see additional information is found in the three-dimensional topology of the folded protein, because they have primary structures that fold together, and protein folding we know almost nothing about. And then they form a secondary structure, then a tertiary, And there's even a quaternary structure. And each new structure in the three-dimensional topography adds new information. We don't even know how the things fold, you see. So more and more it gets, as Alice in Wonderland again saw earlier, curiouser and curiouser. And it's as if God is now saying, okay, as skepticism increases, let me bump up the evidence. I'm going to use technology for my source. I'm going to use science as my source. I'm going to use the very thing that leads to hubris, the arrogance of humanity. I'm going to use that as my source of humbling them. But even there, many will refuse to see it. Why? They're going to plump for directed panspermia, as many have done. What does that mean? What is panspermia? Anyone know? Seed, get the idea, speed, okay? Seeds of life. They're going to be actually because of these problems, some writers, not all thankfully, have appealed to what they call this a desperate measure. They're admitting it couldn't have evolved on the earth. So what do they say? It came from outer space. I remember when I was a kid, there was a movie with that title in the 50s. It was dreadful, but it came from outer space. I used to be scared easily with terrible, dopey films. But the idea is it couldn't have, so it had to come from outside. What's the problem with that solution? It's really not complex to sort out the problem with that. It's not an elegant solution. Why? All right, where'd that come from? How did that evolve? You're just pushing it back one step further, you see, because it couldn't have evolved here. Why would it evolve there? So where did that life come from? And so here you're appealing to something without a designing agent, see, and you have a real problem. See, you're appealing to something like this. And so it just doesn't work for you, you see. So given this notion here, you've got all kinds of possibilities that don't make any sense. And so I use as an example rock cairns, just as, let me, bear with me and you'll see what I mean by this, because this is an interesting way of approaching this. So if I look at these, let me just turn this off here and go to here. I'm interested in these because we've done this a little bit before, but they're fascinating to me. Again, you go into a riverbed or a stream and you see something like this, you get suspicious. You're pretty well sure that this thing doesn't just form there of its own accord, or that some kind of forces of nature was responsible for this, because you see there's some obvious goings on here. So here's another example of one that's pretty. Some people really have too much time on their hands, you see. And here's another one. That's a really elegant one, isn't it? I mean, somebody really had a lot of patience to put that guy together, you see. But you notice I'm saying somebody, not something. Yes, it's possible that it was a chance probability, but I wouldn't put my money on it, and no rational person would. And yet, here's my point. This is simple in comparison. It's child's play in comparison. Let me go off of this just for a moment, because a thing that comes to my mind from another book that I like very much. Let me get it here. There's a book called Pheromone, and I love this book because it's the insect artwork of Christopher Marley. And what's fascinating about this book is that he kind of gardens the beauty of beetles and butterflies and so forth, you see, dragonflies. And so he actually puts them together in this manner. And so you can see that. Now immediately you see this. What do you observe when you see something of this nature then? What do you conclude? Well, immediately you see, oh, there's a pretty clear pattern. Blue, 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 blue. It gets you rather suspicious, doesn't it? That somebody made it that way and that there's an order. This is never going to appear in nature, will it? You'll never see a collection of beetles just in that order, would you? So what do we conclude when we see that? What do we say? Somebody, that's design. Somebody made it that way. Would we all agree with that? So we have that. That's not a problem with that. But yet the amazing thing is, any one of these beetles, though, is infinitely more complex than this design. And we attribute that to chance. Do you see how this works? It goes beyond all reason. And yet this is what we've trained ourselves to do. We're blind to what's right in front of us. It's staring us right in the face. And so again, we see, oh, obviously somebody put them together like that. But we take the things themselves as if they were ordinary and that they just could appear this way. So it is also with butterflies. And you see, again, these complex structures. And so I find it fascinating when I look at a book like this, that you can see that these things here, yes, it's obvious that somebody put them in this order. You'll never see them this way ordinarily, but you look at the beauty within that. And that's what speaks of the glory of God. Now, I believe, by the way, this is anticipatory. You and I, even now, are gardeners. We garden what God's made, but the things we garden are beyond our comprehension. You can't understand a leaf, you can't understand an insect, let alone a bird, you see. But we garden and we cultivate, we curate. We'll be doing that even in the new creation, in the resurrection, and in the new earth, in our new bodies. We'll always be doing, I sense that there's something going on that goes beyond human comprehension. Who would have thought of this marvel, this device? And the more we look at this, the more you study it, the more elegant it becomes. And God's play and his variety, his imagination is mind-boggling. Now back to the rock cairns. That was my little diversion there. So we go back to these. And some people get very elaborate. A lot of people have a lot of time on their hands, you see. This Pontus Jansson, though, is amazing to me because he's got videos of how he's made these things. He actually balances these things, you see. So he actually has videos of how he makes these things without any materials. He has a genius for angling these things and figuring out how to make them, and he adjusts them until they fit. And they don't last very long, but they do stay there. And he takes photos of them like this. So again, if you were going to go out and see this, you'd be pretty confident that this is something that somebody put there. Some are very, very beautiful again. But again, when you see these marvels that he's made and compare them, you realize there's something going on. But I think he's in Norway, I think, yeah. But again, going back to this, what do you observe here? Here are butterflies. We look at the heart thing, and what we see there is somebody put them this way. But our natural instinct is to overlook the obvious beauty that's found there. You see where I'm going with that? So it's amazing to me when we see this wonder and this elegance and this artwork that God has created in his world that's there for us to behold and to marvel and to see. Just to go back to this, though, just for a moment. So find it for you here, rocks here. Here's our friend here. So here is where Jansson does this. So you'll see how he does this. So you see him picking a thing up here. He's a rock balancer. So you see him, there's a video of him doing it. See, and then he leaves it there, and then he builds it. That's one. Then he builds another one like this. And so you actually see him putting it together there, you see. He has a lot of time on his hands, doesn't he? But he does them very well. And then he leaves it and so forth. So it's quite amazing. So he's made hundreds of these videos, you see. And so again, but I use it as an illustration of how we create things and we admire craftsmanship and wonders. Let us then turn to the greatest source of craftsmanship, the greatest source of wonder. Let's turn then to the one who made all things and look at his design and his craftsmanship. So when I go to this, and if I look, for example, at the craftsmanship of the living God, and we're really working on lots and lots of wonderful things here. We've been doing various books I'm having made. It's a PDF, so I can show you these marvels and these wonders that God has made, and in greater detail, so that you zoom down and you look at the fine structure of this creature and the symmetry and the elegance of these works of art and works of wonder that we can only barely and dimly begin to grasp of the glory of God and how this is like a leaf here, you see, and the symmetry and the beauty and the pattern and the wonder and the elegance. So I'm seeking to actually use digital technology to lure people, to romance people back to the analog world. You see, that's what I want. I want to bring them back to the real beauty, which is not AI. We gush about our growth. It is something that it pleases him to make. So I've been doing this on orders of magnitude, on the micro level, on the mini, and on the macro, you see. And I'm doing this because I want to point people beyond to the living God who made all things. And that's really what I'm seeking to do in this presentation as well, to bring us to that. So our next feature is going to be then information theory. And that's going to be fun next week to explore that together and see how God has wired and made us. But see, wiring is a crude metaphor. Your mind isn't a computer. That's crude. It's like saying your body's like plumbing or electronics. We use our own technological ideas, but they're vastly inferior to what he's got in mind because it goes beyond our grasp. And that's the God I want you to know and worship and love. Lord Jesus, we give you thanks for and praise for who you are and what you've made us to be. Amen.
Thanks, kid.